website we need to forge on. And our next speaker will be Arlie Montalvo. Arlie received her PhD at UC Riverside, and since then she's been doing postdocs and a lot of work at different places like UC Irvine, the Forest Service. She's done a lot of large habitat restoration projects, seed germination work, different genetic, she works on genetically based local seeds for restoration, and has published numerous scientific articles. Arlie presently has served as the plant restoration ecologist for the Riverside Corona Resource Conservation District, and is very interested in habitat restoration work, and will be discussing how we can go about the genetic considerations for restoring chaparral, and she's worked a whole lot with California sage scrub. So she's a good expert. And Arlie, can I have you come give your presentation?
Diploids and tetraploids and phylogenies, because I know that you'll just go to sleep. So I will try to make this very general. So primarily we're concerned with how well plants do in a restoration environment in terms of the long-lasting populations into the future. And to do that, we need to consider certain things. And no matter where we plant in wildlands, whether it's chaparral, California sage scrub, riparian, iris, and so on. So, genetics are a cornerstone of many things that we do. They form a cornerstone for managing biodiversity. And genetics form the impetus for a lot of things that we're doing in terms of preserving migration corridors, restoring dispersal linkages among fragmented populations, providing the raw material, genetic variation, within seed sources and plants that we put into projects so that we give those populations the ability to potentially evolve as we have our environments change over time. We're trying to preserve essential species interactions where genetics can be really important. And we're trying to preserve natural processes at all levels, from the population level up to the landscape level. So careful choice of species and seed sources for projects can hopefully provide appropriate genetic variation for projects, help track environmental changes that are going on, reduce adverse effects of inbreeding, minimize detrimental introductions, and just a whole slew of things so that we can hopefully preserve and increase the long-term success of projects. So, why genetics matters. First of all, I'm just going to go over some of the physical and biotic environmental things that are out there that influence plant variation and distribution, because that's pretty critical. I'm not going to get to step 4 in this talk today because there's just no time. Sometime we'll do a workshop on methods for going of how do we achieve selection of populations. So California, as you all know, is an incredibly diverse place. We have the influence of the Pacific Ocean from the west. We have the continental climate to the east. We have the Sierra Nevadas. Is this clear? Oh, the Sierra Nevada range, the Great Valley, the Pacific ranges, the transverse ranges, the peninsular ranges. All these, this amazing topography really leads to a lot of incredible east-west variation across the landscape in lots of things, geology, temperature, rainfall. So for example, precipitation in California varies from the averages of 2.5 to 125 inches. And that's a lot. And even within the belt where we have chaparral vegetation, we have a huge variation in annual precipitation normals. And then there's variation, mountains, the South Coast mountains and valleys. We're going to take a closer look at that. If you just break that down, you can see that we have, even within that little area, and we're sitting within this, we have quite a bit of topographic variation, 300 feet up to almost 12,000. Lots of variation in precipitation, average temperatures, growing seasons, just within those areas. So as you can imagine, that's going to create a real complex environment for plants, and what plants do well where, and what populations do well where. There's a level 4 ecoregion map that's in progress for California, and it provides a lot of detail, taking into consideration temperature, precipitation, geology, elevation, and these maps, I think, are going to be really, really useful in helping us determine areas of similarity for choosing seed sources. So, we have many plant species of different ranges in their distribution. So, for example, the sort of classic common yarrow, or Aquileia millefolium, is a very wide-ranging species. It also occurs throughout North America and in Europe. This is just a map for California. And you can see that this species is in many different environments. This is one of the classic species studied by Klassen, Keck, and Heisey of the Carnegie Group many years ago, where they established common gardens from the coast at Stanford, 10 common gardens going across a transect in California, all the way into the Sierra Nevada. And they looked at the differences in how plants from different populations did growing in different common garden environments throughout this range. And they found that there were substantial differences among populations in their stature, what they looked like, how they grew, how well they survived. And this is a very common sort of situation. There are many plants in California that have much more limited distribution. So, for example, sugar bush, Rhus ovata, which occurs in our chaparral environments and California sage scrub in certain situations, is a species with a much more limited distribution. And that's a very, as I said, a very common thing for many species in California. We have a high rate of endemism in our area. Even sugar bush grows at a lot of different elevations, 6 meters to over 1,700 meters in elevation, and spans a number of different environments. So you can imagine that there are going to be populations adapted to different conditions, even in this more restricted ranging species. This is just an old study, or relatively old, 1998. Does anybody look at studies in the 90s anymore? So it's a physiological study where the researchers were looking at the difference in Rhus ovata taken from a relatively warm site compared to a cold site, not very far apart from each other, and also Melasma lorina, that were called Rhus at that time. And they found that there were differences in these populations in terms of the susceptibility to freezing. And so significant differences among populations and also among different times of year. And so some of this is potentially due to plasticity in the plants, but there's also potentially some underlying genetic influences. And to see that, one needs to study further. But there are indications that there might be something to this. And we need to think about that when we're choosing populations. Traits often vary with changes in elevation and precipitation. Here's Mimulus cardinalis, the scarlet monkey flower, which is no longer Mimulus, as Newman can tell you. However, it grows from sea level to 8,000 feet in elevation. That's a pretty big span. And as you can imagine, it probably has some differences among populations, and it does. So, for example, the triggers that initiate flowering are different for the lower elevation populations compared to the upper elevation populations. And so flowering occurs, the plants bolt and flower much earlier at the lower elevations. And this is while growing grown in a common environment. And this is, timing of flowering, timing of leaf flush, these are all very important traits when you're talking about differences in when freezes occur, for example. And it's very well known in trees that these are adaptive traits. Traits also vary with lots of other things. We know that soils can affect where species grow and differentiation in species. California poppy is one of our incredibly variable species in this state. It's our state wildflower. It's the Schultzia californica. And toward the coast, we have evergreen perennial plants. They survive year after year, flower. They have larger seeds and no seed dormancy. As you move inland, you start to get to annual populations. So finally, you're in the hot, dry interior areas. You have plants that are obligate annuals, so they grow from seed, they reproduce, and they die. They can't parenate. And they have high rates of seed dormancy. Can you imagine growing that evergreen perennial population, or trying to, that's adapted to the coastal, sandy, humid environment in the interior valleys? This is the same species. What would you think would happen? There are ways to start looking at what happens when you start moving these populations around to understand whether there are underlying genetic differences among populations that are going to affect how well they do when you plant them in the environment that you're concerned about. So experiments can reveal whether there are adaptive differences that might affect translocations. A very traditional experiment is the common garden experiment. Provenance testing uses common gardens. These studies can detect whether there are underlying genetic differences among populations. And there's just a very simple, basic model. What you do is you take a lot of individuals from a number of different populations and you grow them together in the same environment. So what you're hoping for example, in this experiment with Nacella pulchra, purple needlegrass, planted in a randomized design, is that the total variation that you see in these plants would be equal to the variation that's due to the underlying genetics plus the variation that's due to the environmental influences on what the plant looks like or how it behaves, the phenotype. When you grow things in a common environment, you're diluting all that environmental variation. And so what you see are primarily the differences due to the underlying genetics of the population. So it's a very simple model for being able to detect differences. One can then take these sorts of common garden experiments and place them in different environments. And so you might see genetic differences, but do they matter? Okay? When you start putting these populations in different environments, you can start to detect if there are adaptive differences to some of the traits that you're looking at. So, for example, here's environment 1, and we have, this is representing many plants, say an average of a yellow flowered plant. In environment 2, maybe it gets smaller. Here's a plant from a different population source growing in environment 1, and it's doing pretty well in environment 2. Here's a population of plants from source 3, and maybe they croak. Okay, maybe that's the poppy from the coastal environment placed in the inland environment. When you see these sorts of switches, that's really good evidence for there being adaptive differentiation between the populations that's under genetic control. You can also see that sometimes populations look different in one place compared to another because there's plasticity to how the plants react as they're growing. And plasticity itself can be adaptive. So I'll just tell you briefly about a study that I did while I was at the Pacific Southwest Research Station with Lotus scopareus, or what do we call it, deerweed? Okay, or California broom. One of those that's used a lot in restoration because it does so well, it fixes nitrogen and so on. This is a variable plant. It's called Acropisthan glaber now, no longer in the genus Lotus. In this study, we had source populations from 12 different areas along the coast of California and from inland locations. And we took seeds from all those populations and we planted little individuals in 2 different common garden sites, an inland site and a coastal site, so inland and coastal. And we then tracked their success and we looked at the survival and flower production of the plants from the different populations. We found that there was a significant correlation with how genetically different the populations were from the source sites compared to the populations that were from the common garden site. So the bigger the genetic distance, and also the larger the environmental distance of the source habitat compared to the garden site, the bigger that difference, the worse the populations did on the average. So that was evidence for there being some sort of local adaptation. So we would predict that potentially if you start mixing populations that are really differentiated, that we might start to see problems down the line after hybridization. So the next step in this is acknowledging that species interact. Okay, not only do different populations of the same species interact when you place them together, potentially interact, but also they interact with other species. So here we're going to first look at what happens when you start mixing populations together. Sometimes there can be beneficial effects after hybridization of populations. We know that we can rescue populations from having too few incompatibility alleles, for example. We have a problem finding mates in a self-incompatible population. And we can rescue populations by adding more genetic material. But sometimes the problem, if the populations are too different, we can start to run into adverse effects, such as mating incompatibilities. Maybe the hybrids don't do too well. There are different things that could be going on. So this is a simple model to look at that's just giving us an idea of the effects of inbreeding, where you're mating with close relatives, such as within populations or close related populations, compared to mating among individuals among populations. So this is how well the progeny are doing, and putting babies into the next generation, along a scale of the genetic distance between the parents that are involved in producing progeny. And so we get an increase in fitness in the beginnings because we're getting rid of inbreeding depression. Inbreeding depression is something that's very, very common in nature and something we really need to avoid. Often much stronger effects than outbreeding depression. But as we start getting too different, as the populations become very different, we can expect that we might start to have some of those irregularities occurring. So that's where we might run into what we call outbreeding depression, where the hybrids don't do as well as the midpoint of the parents. So what we did in this particular study is a bunch of hybridizations between 6 different populations of Lotus scopareus, including within population crosses and then all possible combinations among populations. And then we counted the seed production, we took those seedlings, we planted into the field. A couple of people are here that are actually in this picture. We have Bonnie and Jenny, who are sitting in the audience. This is a long time ago, when they were babies. So planted them in 2 contrasting gardens, and we looked at the cumulative fitness of the populations involving how much seed was produced from the crosses and how well the seedlings did in terms of emerging from seeds, how well those survived in the garden, and then the flower production of the surviving individuals. And we found that there were significant differences, in how well these hybrids did, depending on how genetically different the parents were, the population crosses, and then also how different the environments were of the parent, the average environment of the parent environments relative to the common garden planting site. And the environmental distance really trumped the genetic distance in this case, showing that there was, I keep pressing the wrong button, there was a real importance of the environmental similarity in this case, even more so than the genetic similarity. So it's just, so there is a scale of the differences that really mattered. So we also need to consider community interactions, not just the interactions among individuals of the same species, but what about local adaptation to soil organisms? Are there differences in interaction of different flower forms and flower colors with pollinators? There are many different interactions to take into consideration when you start moving populations around. There's also the background environment. What was the competitive situation where plants were growing compared to where they were, compared to where they're being put? We have found that studies in Nucella pulchra and Elymus glaucus, 2 bunch grasses in California, Rice and Knapp found that depending on where the source populations, that source populations differed in their response to competition, depending on that you could see the difference in local adaptation more strongly under competition. There's some other studies where people have looked at how well populations have done from a competitive environment compared to a non-competitive environment. So there are lots of different ways to look at this. And I don't have time to go over genetic assimilation, but we also have to be concerned with those sorts of different species interacting, planting common species next to rare species, and potential hybridization, absorbing rare species populations. When we know about this, Ceanothus is a prolific, outcrosser. You get a lot of hybridization. So there are lots of different species where this could be potentially a problem. We have also had the situation where hybridization between introduced species and a native species ended up creating a new hybrid, very aggressive weedy species, such as when Spartina alterniflora was hybridized from a restoration era with Spartina foliosa, and the hybrid basically took over, swamped the native Spartina foliosa, and created a huge weed problem in the San Francisco Bay. So we have the little horror stories that we should always keep in mind as well. So just in summary, when doing your thought process for where are you going to get your seeds from, and where are you going to get your stock material for growing plants for restoration, if you don't actually know a lot about the biology of the species, just try to collect in a more, at least regionally local matching environment sort of situation. If you can learn more about your species, that's great. There's a lot of information that we can put together and pull together to make sensible choices. We also need to think about climate change, and we need to think about how do we move up or down in elevation or across gradients, but carefully, not just randomly, in terms of how do we seed, maybe pepper populations with genotypes that might help the restorations make it into the future, especially in this land of fragmented habitats. And I think that's the end.
we're happy to be back at the discussion session, and I know we'll have many more questions with all this wealth of knowledge you've got about this. Moving right along, our next speaker is Bruce Berlin. He's a resource ecologist and horticulturalist for S&S Seeds. For the past 22 years, Bruce has worked with project planners, biologists, landscape architects, etc., in an effort to use the right plants in the right places. He's an active member of the Society of Ecological Restoration, CNPS, and the International Erosion Control Association, so he has a wealth of information and some specialties we all want to hear about.
Thank you, Marcia. Really, it's an honor to be here. We have probably the single most collective intelligence on Chaparral that I can certainly think has been gathered in recent times, so including international guests. So that's awesome to have all the components come together. So thanks for letting me come. And Arlie, as always, she has a ton of information and a great lady to follow. So thank you for that introduction. When Marcia asked me to, can you hear me all right? I can't, okay. She asked me to come speak at a Chaparral restoration, and my first reaction was, what? We don't seed after disturbance in the Chaparral, especially when it comes to fires. And so, I'm going to say that offhand. First comment I'd say is, as a general rule, and conspiring with all the things that you've heard so far, as a general rule, I would almost say that your first reaction after a fire isn't to automatically think you need to seed. But we'll go through some of the thought processes about what you do, and when you do seed, and where you seed, and how you seed, and so some of that, and how it relates to what our company does. And so I'll give you a little bit of history and background on that, and I think I can figure this out. So when it comes to considering whether to seed or not, there's basically three basic options, right? You either seed, you don't seed, or maybe you seed in very select areas. And so that's what it really comes down to, is those three things. And so I saw this last night on a BLM slide, and it's a pretty interesting one, although I see a big typo right on top. Persistent isn't spelled that way, but there's a huge disconnect between those that desire native plant materials and those who don't believe it will ultimately give them the results they desire. So you've seen a lot of scientific graphs. This is my 4-quadrant graph here. And so you have the glass half full. I think I can figure that out. The glass half full, they're saying all natives all the time. That's what we're going to do. We're going to put back the natives. Well, those are what we call the tree huggers. And they certainly have their place and their arguments about preserving the native plants and helping them out wherever we can. Then you go down to the other extreme, of course, where the glass is half empty, and say, oh, those natives are so expensive, they never work, why are we troubling ourselves with that? And then, of course, I find myself gravitating. When I was young and naive, and I was trying to make an impression, I started up there, and now I'm definitely down here in the it depends category, where it's half full, half empty, well, it depends, And then there's the other group that says, hey, I ordered the cheeseburger, I don't know what you guys are talking about. So hopefully I don't gravitate towards that other one. So quick show of hands. How many people actually think that seeding does not work? And don't worry, I'm not going to laser you if you say that. That's the goal. Well, okay, so it's not a fair question, but okay, so some people absolutely say seeding does not work. I've spent my money, I've tried, frustrated. Why do I throw my money away? There's certainly a camp that believes that. How many people actually think seeding can work and does work? Okay, and I'll go through some of the studies that have been done. Jen and her people have done marvelous work looking at stuff and seeing what kind of results we can get on seeding project. And I think I would say number 3 is where I fall, which probably the majority are. Sometimes it works when the conditions are right. So going back to the don't seed, well, that's the easiest thing to do. And the lady from Los Padres talked about that. Sometimes inaction is actually an action we take. It's the easiest thing to do. Say, well, you know what, this is nature. It's going to come back on its own. There is pressure with doing that, though, to not interfere with nature. You get the people, again, the people in that one camp that say, absolutely don't do anything. And especially, as you've seen so far, especially with the resiliency of the chaparral, that's often the case in post-fire situations. You don't need to reseed as long as you have a decent enough base of plant material that will come back over time. And then, you know, the obvious thing is it costs money to seed. Why do I want to throw my money out there if it's not going to work? And then there are studies that show seeding does not work. In many cases, you're not accomplishing any erosion control. You're actually not doing anything to help co-generate the plant habitat that you're trying to preserve. So, and I say really, because there's some other examples out there too. Obviously, if you do seed, what is vegetation going to do? Obviously, it stabilizes the soil, minimizes your erosion, allows the water to infiltrate instead of running off and causing the flooding and the expenses with flooding. Vegetation stabilizes the soil for many years. So actually, I think of it as a placeholder. It's really holding things intact until that plant material can come back on its own. So if anything, it can be a temporary stabilizer. And then you've heard several people talk about where they're using now, you know, vegetation, be it seeds or containers, to prevent type conversion, to try to reclaim some of that land we've lost. And then again, seeding methodology, you know, we used to just aerial drop things out there and think that it would work. Well, you know, we've got improved technologies. Hydroseeding has, you know, come a long way. It's still not the best way, but imprinting you saw and pitting. So there are better ways to seed that we used to not ever have available to us. Okay, so seeding in select areas where imminent threats to life. This comes from some of the BEAR criteria that's in the BEAR handbook. So I'd never heard the term BAR yesterday, so that was a new one for me. But I would say that this stuff is, these points are important in some of the criteria that you should look at where you would consider seeding. And I'll just go through these fast because I got a lot of slides. Isolate specific areas where you actually have some threats to either life, infrastructure, or habitat. Use the right species, as Arlie talked about, and even down to subspecies. And you should really try to use as locally available material as possible for the obvious reasons, but also for bigger implications like Arlie's talking about. And then, you know, I have to also give some realistic time frame for how long it's going to take to get some success from seeds. You've heard the dramatic swings we have in rainfall, and I think if you look at a big picture, when I think of seeding projects and why they don't work, it's typically two things, and that would be water or rainfall and weeds. Those are why we lose the battle probably 90% of the time. So basically you want to use, you know, smart decisions about when you're doing stuff and what you're using, and then also have the monitoring and time frames of measuring how well that your objectives have been met. Just the basic diagrams of all the good things vegetation does. I don't need to get into that. Obviously, raindrop impact, infiltration, all the obvious reasons why vegetation is a good thing. It is truly, you know, the best management practice you have as far as keeping soil intact. Chaparral is resilient. This is from some Achilles work early on, and the fire cycle and how it actually, and coastal sage scrub. Now I'm learning that term. I never heard it before today. California sage scrub. It is characterized by herbaceous phase in the very first season, wet season after a fire. Key being wet season. If we don't have the rains, then typically you're not getting much coming back at all. Many of the native herbs are adapted specifically to the fire cycle and can survive. As John talked about yesterday, we always think of fire severity as being hugely important. It's actually the fire frequency, which is much more important. Some of the characteristics of chaparral, we had a wonderful botany refresher. It was really cool yesterday. Enjoyed that tremendously. Some of the dominant chaparral species only come from seed after a fire. And then, you know, specialized annual floras that we have as well. The obligate resprouters, the obligate seeders, the endemic fire followers, the facultative seeders that actually resprout and seed, but the seeds are enhanced with the fire. And then the frequent fire followers, the annuals, the stuff that we only see after a fire. So those are kind of the five basic plant material recovery methods after a fire in particular. This, to me, is still probably the model that best fits in. You recognize it. It's Todd Keeler's Wolf's diagram about post-fire emergency seeding and conservation, and I'll break it out into individual components, but here you're talking about a fire in the wildland on the upper left, and then the thought matrix and the selection about where you would consider seeding. Of these final 8 to 10 boxes, there's only 3 or 4 that actually advocate seeding. So again, and I'll get into some of the specifics of that. Here's an example on the left side of the screen, and I'm sorry I keep going this way, I can go this way too. Oh, I can go this way too. This left side, this is a moderate fire on the left side column, split down the middle with a high intensity rainstorm. you have a moderate fire with some ground covers, consumed, and then you have exposed hydrophobic soil. One of the characteristics of soil, and I was talking to Ted St. John just briefly, some of the characteristics of the chaparral soil are such that it actually does create that layer that actually repels water, so that makes it even more difficult to get plants going. In those cases, with the dry gravel and things like that, you get overland flow, soil erosion, loss of nutrients and seeds, and it leads to flooding and problems. That's the left column. And then on the right column, where you have a lower severity fire, you can't get that to work, but that's coming back over here, sorry. Low severity fires where the ground cover is still intact, you actually probably have some protective soil cover, you still have some infiltration, and your soil, seed, and moisture is still present, so your plants are probably going to regenerate. And so that's a model from, I've got to give credit where due, Jordan et al., post-fire wildfire soil erosion from the Wildland Urban Interface Symposium in 2008. Pardon for not citing that earlier. So here's, we talked about a little bit yesterday, the burn severity and some of the temperatures. It's really interesting to hear that 250-degree subterranean heat gradients. Well, this is some of the analysis that had been done, and this is from Hungerford and Ivano in 1998, 1996, where you have 3 different severities of fire, low, moderate, and high in the 3 columns. And, oops, I'm going to learn that yet. The key is that you get to soil temperature at 10 millimeters or just about ½ inch, greater than 480 degrees, in that high fire intensity, and soil organisms down to 6 inches could be cooked. That's pretty tough. I mean, because then the soil biota, which is so important in helping some of these things regenerate, either seedlings or sprouters, it's cooked off in some of those intense fires. But and some of the low and moderate ones, not so much. And it sounds like John's maybe doing some additional work on that. I don't know. So history of post-fire seeding, what has been done over time. Most of you know this or have seen this story. Back in the 1920s and 30s, they actually started out with a noble cause. Way back then, they were actually doing container planting of stuff after a fire, much as you saw in the Angeles Forest doing today, with some of their restoration efforts, where they get results. They need to have the percentage of cover, so they used to do that, but it was only truly by containers. They weren't really doing the seed planting at that point in time. A little bit of that. And in the 30s and 40s, we see some of the bad things that we see still today, the annual grasses, including such crazy things as barley and mustard. And I hate to say that, mustard used to be one of the things that they plant. I mean, as crazy as that is. And the dormancy on that, of course, of that seed is incredible. It'll be around for years and years. And then as we move toward the 40s and the 70s, annual ryegrass and some other grains you start to see. And then more recently in the 1980s, some of it by a federal directive to use native plants. They start to use some, and now we're seeing more, of course, just mulching and no seeding at all. That's more typically the case these days. Very little seed being used in California especially. One of the studies that I allude to that we looked at some of the effects of seeding over time, and Jan and her people, and that's Peter and Neri and these other people here. This one was evaluating post-fire treatments, and one of the things they looked at was, so they actually looked at 470 fires, 321 bear projects for 25 years, from 1973 to 1998. So it's a little bit dated. This was published in 2000, I believe. Yeah, 2000. But it was looking at some of the early stuff. And bottom line is they were trying to evaluate how much money is being spent and whether it's effective or not. What it turns out is that 91% of the ground seeding efforts, meaning that they didn't aerially apply it, they actually have to be excellent or good. 81% of aerial seedings were either fair, good, or excellent. But seeded grass could also do other good things, like maybe keep out some of those noxious and invasive weeds. And seeded plots actually ended up having greater cover, about a little more than half of the time. So this comes looking at Region 5, which is our California region. Of course, they looked at 32 different events, and they ended up having pretty good response, where basically less than 1 in 5, about 20%, just under 20%. They got poor results, and everything else was from fair up to excellent, but most of them falling in the fair category. So this is some of the research Jan's been doing, which is great. She's helping other people out in different regions too. Rapid vegetation establishment has been regarded as the most cost-effective method to mitigate the risk of increased runoff. Well, they still think that plants, obviously, getting that plants up will help minimize some of the runoff. And that's from something that Jan did in 2004. Federal policy, again, mandates the use of seed from native species or post-fire rehab whenever it's available and economically feasible, which are two key words there. Has to be available, and you heard somebody mention that seed availability has never been very good, and it's also expensive. And then, although the use of native species has increased, high cost and inadequate availability. So a little bit of that there. And then this is some of the results they had, and so we need to start to work together as a seed industry. And I just came from a native seed conference earlier in the spring, National Native Seed Program. And it's a really neat program that there's 25 or 30 different seed producers and collectors that are actually working with the BLM to get the right species from the right areas. And so I'll give a little example of some of that is, some of that, how that program works. Basically, sediment production's increased. I'm just going to fly through some of these because I got too much to cover, and I didn't start my clock. Okay, so bottom line, you're looking here, obviously plants with greater cover. On the seeded stuff, you actually ended up with pretty good seed cover. On almost all of them, the coverage range that seeding sites actually had increased coverage anywhere from 11 to as much as 40%. So seeding can work, okay? Where is seeding appropriate? This comes directly, I pulled the information off of Todd Keeler-Wolf's slope erosion. They're looking just at slope erosion, and this is post-fire examinations of sites where you have unstable slopes, where slopes are directly threatening some structures or some other sensitive resources where you're prone to dry rattle. Well, then maybe you consider some seeding. That's one of the areas you potentially consider using some seeding. Fire in a wildland, again, with some structural or resource threats, you do the vegetation recovery assessment. If you don't think you're going to have a very good return because of maybe a fire intensity is too high or the fire frequency is too high, and again, you have some threat below, then maybe again you look at using some native seeds. This is again fire frequency, so it's not only fire intensity but also fire frequency leading to that. So those are the two situations. And then of course the timing of the fire, whether it's early or late, that also has a huge factor as to how resilient your recovery is going to be on its own. Soil evaluation. I question whether, and I didn't hear you talk to that in the BEAR analysis, how much of this analysis is actually done to evaluate what your seed bank is. Do you even know what your potential recovery from seeds is? I know it's a cumbersome task because you're talking, in some cases, you got to go 3, 4, 5, 6 inches deep to get below that hydrophobic layer because that's probably what's going to be what's sprouting. But I don't know, it needs to be done for certain, and it's not an easy task. But seeding rates will vary all over the board. What it comes down to is you need to know seed counts. Typically on some of the drill seeding and stuff, they look at 40 pure live seeds per square foot, which sounds like a lot, but you're going to have some mortality there too. I'm going to fly through some of these. I want to just show you some of that. So some of the criteria in seed selection, how do you choose? Well, certainly you got to work with what you have to begin with. You want to find out how quick it's going to establish, how compatible it is with your other resource objectives, and how adaptable it is as well. Native versus non-native. Well, I don't even think we need to discuss that anymore. I'm going to say there's a lot of native plants that are now available, be it annuals or short-term perennials, that will do the same thing we were looking to do in the old days by using ryegrass and some of the grains. And that would be things like the sterile triticale, the sterile wheat times wheatgrass, which may produce one crop, but that would be it, and then you'd be done. Or look at things like native annuals, Vulpia microstaches, Muhlenbergia microsperma, some of the native grasses that are short-term and passing. Seed testing, I will say, if there is ever anybody buying seed, you should always ask your supplier to give you the information from a current seed test so you know what the seed count is. You can do the math and figure out how many live seeds you're dropping a square foot. But more importantly is that you see the weed seed and know what else you're bringing in with that seed. It makes me a little nervous when I see straw bale dumping because there's definitely some weed seed or seed in there, and that's another thing that should also be tested. And don't use TZ tests. I don't know if you guys know what that means. That's tetrazolium, and that's just a dye they put on the seeds that show the potential of that seed, meaning that it looks intact, it absorbed enough dye that potentially it should grow. it's really not a very accurate way to go, so you should not use tetrazolium tests. Seed zones, already talked about this some, and it's really important to know elevation changes are huge, and so are also inland and coastal changes. So I don't know what the right range is, 500 miles. I've seen it as high as 20 miles. So the closer the better, I'll say that. Here's just, I'm sorry I got to get this in. This is the BLM model, and there is some people from BLM here, but they actually have a plant program and seed bulking program in place. It's kind of interesting, and I'll just walk through it real quick. 6 steps to it. Native seed collection. You start out by getting some seed collection. This is called the Seeds of Success, the program. Some of you are already familiar with it. And so you go out in very small quantities and get perhaps a lunch bag full of seeds. Then they can grow it out and evaluate it and develop it. But they do go out and have it all GPSed, and they know where the plant material and stock material came from. Then they evaluate and develop it. They grow it out and actually see how it expresses itself. And they'll send that, and they'll do that through the Plant Material Center. And then they will do field establishment. We'll plant out rows of it and see how it produces, see how it performs, see how it responds to stresses, things like that. Step 4, then they actually will take and make some of that stock seed available. We're currently growing 2 different NRCS releases. One of them is a Muhlenbergia spirifolia, and the other one is Spirobolus areoides, called Vegas, and it's from the inland areas. Beautiful, and also it's nice to know that we're helping put back the right plant in the right place. Then seed storage, BLM actually has a warehouse where they keep seed in stock, ready for fire. So somebody had, I think Marty or somebody had the example of the fiscal year, and here we are in June, and they have no money left for the rest of the year. And the BLM probably gobbles up a lot of it. They've already bought it all and sitting in their warehouse. So somehow, Forest Service needs to get to that table and make sure they're getting some of that pie too. And I don't know how you intercoordinate agencies like that, but the DOI or whoever all the players are needs to come together as a whole and try to work together, because it's a shame that, and I'm not saying it's in seeds, I'm just talking about everything. All the resources need to be shared. Step 6, it goes back out onto a project that's using and looking for that. Seed collections. Okay, my time is up. I will cut it short. I'm sorry. We don't do seed collections willy-nilly. We got to go out. If we're working in Forest Service, we have to get permits. Part of the problems about making seed available is typically we're not allowed to go in post-fire areas that are recovering and get you those fire followers. So bottom line is we try to be ethical. We occasionally getting into trouble, but if so, we give you back what I've been on state parks land. I didn't even know it. I had to give them their seed back. But anyways, we do what we can, and seeding is certainly an opportunity for you to look at, but it's certainly not a cure-all, and especially post-fire. It should be a small part of what you're doing. Thank you very much.
We definitely will have questions for Bruce during the open discussion. Okay, our next speaker will be Edith Allen. She's a professor of cooperative extensions. She's a professor and cooperative extension specialist in the Department of Botany and Plant Sciences at UC Riverside.